Very much, Nancy, and I want to extend my appreciation to the ISTC for holding this symposium. I think it's a very good idea. 3 years ago, when I started in this whole biochar research area, I did a search on Google. There came up to be about 1,200 web pages mentioning biochar. I just did it this morning again, and there's 850,000 web pages with the word biochar in. This has been an absolutely explosive field. There's very few examples of something going from a scientific realm out to public literature within 2 to 3 years at this volume of material. So that's something to keep in mind in all this. So before I get going with the presentation, I first wanted to give you a little background on the USDA ARS. I am a member of the intramural USDA research team. However, we're not only just the USDA research team, we are each of your research team as well. We have about 100 locations across the U.S., and you can see I have a couple of the various multi-location projects specified, as well as the Biochar Paralysis Initiative. Currently, there's 15 USDA ARS labs that are working on biochar. And you see that I have it referred to as the Biochar and Paralysis Initiative. We are not quite yet a project because we don't have a source of funding yet from Congress for this, but we are an initiative. Within that initiative, you see the purple double-boxed sites. There are 6 locations across the U.S. that are utilizing a fast paralysis char in field plot studies. The purpose of this is to actually see what occurs in the field with the same char, the same crop, as a function of the different climates and soils at those different locations. So what I was going to present to you today is a little bit about the biochar's interaction with the soil microbial communities. But first, I'm going to have a little biochar minus 101. I appreciate Katie's presentation as well as the video we saw to begin with, but I'm going to give you a little different view on all this. In your folder today, we actually got a pencil, and it's one of the nice original graphite wood pencils. We as humans have been using charcoal for over 35,000 years. This material is very unique. Just think about all the things we use day to day. How many of those materials have been around 10 years, 100 years, or 1,000 years? Yet our charcoal has been around for 35,000 years. So the overall process of paralysis, carbonization, coalification, the process by which we get carbon, is well established. They have well-documented research histories. The one problem has been, all of the emphasis up to today has been on fuel production, primarily on bio-oils. The solid, what we are calling biochar, has been considered a non-desirable byproduct. Basically, it was lost energy. If you ended up with a high carbon residual, you weren't converting things efficiently. So what has changed in this whole process? Well, the whole idea of biochar is on the purpose of the creation of the charred biomass. It is to reduce atmospheric carbon dioxide levels. So the whole idea of biochar is a means of atmospheric carbon sequestration. This idea dates back only to about the early 1980s, and then it really catched a lot of momentum in the early 2000s. So we talk about carbon sequestration, and what is that actually? It's the storage of carbon in soils. So I show up here a couple different ecosystems. The top is the agricultural croplands. On average, we store about 0.2 to 1 ton of carbon per meter squared or per acre per year. The forest, we do a little bit better, about 0.1 to 4. Grassland and prairies, we do about 0.1 to 1 tons of carbon per acre per year. Swamps, floodplains, and wetlands are our best places right now for carbon sequestration, and we're doing about 2 to 4 tons per acre per year. These numbers, there's a lot of variability, but these are the approximate ranges. So our whole idea of biochar is we're going to try to increase these rates of carbon sequestration. The one thing I like to note is our highest sequestering ecosystem is also the ecosystems where we're anaerobic. So you can kind of see here that our rates are directly proportional to how much oxygen we have in our overall system. That shouldn't come as any surprise because microbes love converting carbon. So I said that biochar is more of the name given to the process. So what is the solid residual? It is part of what's referred to as the black carbon continuum. And what you see up here, we have a big problem with how we refer to this black carbon. Jones, back in 1997, saw this problem in all the black carbon literature, and he proposed the following guidelines. If we look at the oxygen to carbon molar ratio, what Katie was just presenting earlier, we see that graphite has a ratio of 0. There is no oxygen in that structure. The next material is soot, which Jones defined up to about 0.2. Then charcoal is from 0.2 to about 0.5. Char is a name given to materials from about 0.4 up to about 0.6. Biomass usually has a ratio of O to C greater than 0.6. So what do we actually have here? On this side of the graph, we see that we have all our thermochemical conversion products. This dividing line of 0.6 separates our biomass from all our charred products. We have another important dividing line here at 0.2. This is where we separate condensates from residuals. The difference in residues is we actually are able to see relic structures of that biomass. You saw in Katie's SEM slides, where when you looked at the wood, you kind of saw material that looked kind of like wood that you would see it today, except it was in black in color. What we end up with in the soot realm is we lose all those relic structures. Soot is actually reformed carbon material. So it's referred to as a condensate. So what is biochar? Biochar spans all of these realms. We go, the lowest biochar I've seen is about 0.10, all the way up to about 0.62, 0.65 in the O to C ratios. So when we see biochar, it spans all the realms of this black carbon spectrum. It is not a new division in the spectrum. It's referring to the process and why we are converting that biomass. So the other thing that we often hear is a lot of analogies. So what I've compared here is our natural biochars versus synthetic biochars. The natural products have a lot of impurities. When you think of something burning in the environment, we have soil, we have oxygen, we have a lot of minerals mixed in. The problem is these impurities causes changes in these thermal conversions. It's been known for some time that metals cause more solid forming reactions. So what you end up with in the natural biochars is a real mixture of a lot of materials. We not only have impurities, we also have multiple feedstocks. So I kind of show this down in these pictures. When you think of a lava flow moving along, it's going to be burning whatever is present there. Same thing with the wildfire. Now think about our pyrolysis processes. We have a homogeneous feedstock getting fed into a constant temperature reactor. This is an entirely different beast than our natural products. The other factor is what happens to that material after it's formed. The natural products are air-cooled under oxygen. They're exposed to solar radiation. They're exposed to precipitation. When we look at our synthetic products, they're typically cooled under anaerobic states. There's typically no water involved, so we end up with different possible reactions after formation. So if you look at the literature right now, there's kind of 4 groupings for the interaction of biochar with soils. The first is that there's an alteration of the soil physical and chemical properties. We look at pH, CEC, decreased bulk density, increased water holding capacity. All of these factors are important and they do happen. The second is biochar provides improved microbial habitats. I refer to this as us putting up these nice multi-million-dollar condos in the soils. We have this idea that since we're putting in this material, the microbes are going to leave the soil, go over to our biochar and say, oh, this is a wonderful place to live. The third is that we have this overall hypothesis that there's absorption, desorption of soil greenhouse gases and nutrients on this char. The fourth effect is some indirect effects of the mycorrhizae fungi through other effects of microbes. And what Warnock was getting with this is there's other bacteria present in the soil that are referred to as the mycorrhizae helper bacteria that produce furans and flavonoids. These are chemical compounds that are necessary for the spores of fungi to germinate. And I'll get back to those two chemicals here in a second. So overall, these are the four basic mechanisms that have been proposed for biochars. So one of our big problems in the biochar arena right now is when we talk about stability. If we look at the literature, and I've kind of summarized the more recent studies up here, we have residency times going from less than 30 years all the way up to millennia. This poses a lot of difficulties when we look at what happens to biochar in soil. The other thing that I like pointing out is biochar isn't new. Like I was mentioning, it's part of the black carbon continuum. The very first study of biochar degradation was back in 1908. This study by Potter was the first one to show that fungi can degrade biochar, black carbon, or in this case, it was low-grade charcoal. Excuse me. So what are our possible explanations for this? Just recently, I compiled all of the literature that showed stabilization rates, and I looked primarily at the O to C molar ratios. Remember back when I showed the continuum for black carbon? I said that the 0.2 and the 0.6 were important dividing lines. You can see I've included those here. The 0.6 was the dividing line for biomass. The 0.2 was the dividing line for condensates. So what you see is I kind of have three regions highlighted here. We take all the studies from the literature, plotted them on this graph, and from this, I'm kind of proposing that there's three different zones for stability. The first, if we have a biochar that's greater than or equal to 0.6, its half-life is going to be less than 100 years. For these middle biochars, their minimum half-life is going to be between 100 to 1,000 years. And this final category, where our O to C is less than 0.2, we have a very stable biochar, and its half-life is greater than 1,000 years. There is this little red box here that is the study of Harmer in 2004. These were 4 particular hydrothermal biochars that were made over a period of about 24 hours. There is the possibility that there was other reactions that occurred that all these other biochars didn't have, but anytime you look at a range from the literature, to have one exception is pretty good. And so we look here and we have 34 studies that all kind of agree on these dividing zones. So what happens when we actually add this biochar to soil? Well, the first part is we know when we're sick. We don't feel well, we have a fever, we usually stay in bed. What happens about soil microbes? How do we know when they're sick? When I usually give talks to first graders, second graders, and even my little nephew, I say that I often look at bug farts because it puts it in terms that they can understand. And that's literally what I do. I look at the products from the microbes. These boil down to the gases. It's carbon dioxide, methane oxidation, and N2O production. So overall, I look at implications on the rates of reactions as well as the gases that are produced. This allows me to look at some of the clues into the mechanisms of what's actually happening. So what I've actually done in my lab, so far I've analyzed about 44 different biochars. There's been 11 different biomass parent materials, and you can see them up there ranging from the hardwood, softwood, through the corn stover, corn cobs, macadamia nuts, peanut shells, sawdust, algae, coconut shells, turkey manure, and distiller's grain. These samples represent a cross-sectional sampling of all the available biochars. I don't actually have a pyrolysis unit. I wish I did at times, but other times I wish I didn't. So I actually don't, and I'm reliant on other people providing me chars to do the testing. And so this really gives me a feeling for what's available across the board right now in the industry. So we have biochars ranging in carbon content from 1% up to about 84%. The nitrogen contents vary from 0.1 up to 2.7%. We have a whole range of production temperatures going from 350 to 850. And I have a whole hodgepodge of processes ranging from slow, fast, gasification, as well as hydrothermal. So what have I done with all these? I've run various soil incubations. The first type I call my mini incubation. I put 5 grams of soil with half a gram of biochar. I do it at 2 different moisture contents, field capacity and saturated. The other incubation I call my jumbo. Primarily this came out of the idea that I wanted to start looking at what implications biochars had just being the biochar and not necessarily mixed with soil. So I'm not going to bore you with all 43 chars. I would love to, but we don't have the time today. So I wanted to show you just typically what happens when you mix biochar with soil. There's a lot of problems measuring the CO2 production from biochar. One of the issues are carbon itself is not stable. The thermodynamic favored product of solid carbon is CO2. That's why we get energy out of it when we burn it. So the solid carbon technically is not fully stable. So we have to look at the CO2 production that we get if the biochar wasn't in the soil. So in order to come up with the true impact, you have to subtract off what you see in the soil plus biochar from what you get from the biochar alone. When you do that, you end up seeing that when you're adding biochar to soil, you actually get a decrease in the CO2 respiration. Remembering back that this is our little thermometer for how the microbes like the biochar, and we start seeing as we mix more and more biochar in the soil, we start getting lower and lower levels of activity. The N2O is the same story. We have high production initially, but then as production decreases, as we keep adding additional amounts of biochar. The methane oxidation shows a similar trend. As we add more biochar, we get lower rates of methane oxidation. This is important because the soils are a sink for atmospheric methane by this process. So as we're adding more and more biochar, we're shutting down all the microbial processes. So then, remember I told you about the jumbo incubations? I wanted to look at what would happen if we didn't mix the soil with the biochar. If we just put the soil in an incubation vessel and had the biochar in the sealed vessel, but in a little beaker so it couldn't actually interact with the soil. What you see here is that we actually see a similar effect. The methane oxidation isn't as great as the nitrous oxide, but for the methane oxidation and the nitrous oxide, we basically see the suppression whether or not that biochar is intermixed with the soil. So when we saw that, we started thinking, well, what could actually be causing that? One of the things that happened in my lab was we had another system, a gas chromatograph system set up to analyze a bunch of hydrocarbons. The samples accidentally got run on that system, and we saw ethylene production. So that's what we started pinning on as a potential mechanism. Since it was occurring without the char mixed with the soil, we knew that it had to be a gaseous product, because that's the only way that something could inhibit reactions was being a gas. It could get from the biochar into the soil without actually being intermixed with it. And what we see is that indeed we have that. Ethylene was discovered in all the biochars. You see here over on the left, we have the soil and an activated carbon control. We didn't see it from the activated carbons. We only saw it from our biochars. And that, it wasn't a consistent trend. You can see the rates vary, as well as some chars are better ethylene producers than other chars. So we have a whole variety. So when we saw this ethylene, I got really excited because ethylene for the soil microcommunity, it induces fungal spore germinations, it inhibits and reduces rates of nitrification and denitrifications in the soil, it inhibits methane oxidation. So all of a sudden I had a mechanism to explain all of these reduced rates that I was seeing. Furthermore, ethylene is also involved in what's referred to as the flooded soil feedback. What happens when soil is anaerobic is we get higher ethylene production because ethylene oxidation is actually shut off. This involves a lot of microbial and plant responses, one of which is referred to as adventurous root growth. So when you have ethylene present in soil, the plant responds by sending out a lot of fine roots. And this can be seen in a lot of the greenhouse pot studies with biochar. If you pick up the plants, you typically see these slides showing all the fine roots in the biochar grown pot compared to the control. This could be the one mechanism responsible for that. But I want to focus still on the soil today. So what we see in the bottom graphs are the response in the N2O production as well as ammonia production to ethylene. And what you see is as we increase the concentrations, we have decreased nitrous oxide production, but we have increased ammonia production. So what happens in our biochar soils? We see a very similar trend. Nitrate production typically goes down as we add increases amounts of biochar. The nitrite is unaffected, and we typically see an increased ammonia concentration. Now in the literature, there's a lot of disparity saying, well, the nitrate ammonia could be coming from the biochar itself. And these little inset graphs are the extraction of our controls, just looking at the biochar. And you see, yes, there is a little bit on the char, but it's not a significant contribution to this overall impact. We also looked at the desorption phenomenon. Yes, it does occur, but again, it can't explain this 90% decrease for this particular char. So what is actually happening here? So this is a very brief overview of the nitrogen cycle that occurs in soil. I'll start up here at the top where we have the nitrogen uptake by plants and microbes. Once they die, they form an organic nitrogen pool. That organic nitrogen then is transferred to ammonia by mineralization. We can also have ammonia formation directly from the nitrogen-fixing bacteria. Once we have ammonia, that can undergo nitrification by the nitrifying bacteria to go to nitrite or nitrate. Once we have nitrite and nitrate, that enters the denitrification cycle to get NO, N2O, and then finally N2 to start the whole cycle over again. So what's happening when we add biochar? First of all, we see increased amounts of ammonia. The second thing, we have decreased amounts of nitrate. We have decreased N2O production. And I didn't present the data, but we also have decreased NO formation. So from all of this, we're beginning to get a picture. We don't have all of the answers yet, but biochars are interacting with the nitrogen cycle by really shutting off or reducing the nitrification rates. And they could also be reducing the denitrification rates, although we don't know that for certain because once the nitrification is slowed, the denitrification is naturally slowed because they're coupled and dependent on the products of one to be the reactants of the other. So the thing that we can say initially here is nitrification is reduced. So we saw ethylene, and I was very excited about that, saying, well, ethylene could be a mechanism for this whole process. Tim Clough in New Zealand just recently published his view, and he hypothesized that alphapinene was a responsible nitrification inhibitor. And when I first saw this, I kind of scratched my head and was, alphapinene, I haven't heard of this one yet. What is this compound? There's a picture of it. Alphapinene is actually a naturally produced volatile chemical from plants. It is present anytime a plant is growing. It also has been implicated in the overall chemical signaling in insects. So this is a very important chemical through all the biogeochemical cycles. Now, despite our differences in our chemicals that we chose, our overall conclusion is the same. We have the exact same mechanism that it's a gaseous product that's causing the inhibition for the N2O production. So we started thinking, well, if he found alphapinene, we saw ethylene. What other things are present on these biochars? So the thing that we're currently doing now is looking at what I'm referring to as a headspace thermal desorption, GCMs. It's a nice fancy term saying that we're baking these biochars just a little bit. We're only going up to 50 degrees C to see what's coming off of them. In the top graph of wood ash and activated charcoal, you can see there's no peaks. Once you activate the carbon or you have a wood ash, there are no volatiles of detection. The next one down is a wood pellet biochar, and you see that we get some peaks, which I was very excited to see. The next one down is a macadamia nutshell biochar. You see we have more peaks, Additional peaks, the heights are proportional to the amounts that are present in the material. We then go down to the hardwood sawdusts. We even have more peaks. This one was done by a fast pyrolysis process. We then look at an oak hardwood biochar. We end up with more peaks. The distributions are different. Then on the bottom here is just a plain old bituminous coal sample that we wanted to compare it to. So overall, these peak sizes are all in the ppm range. Our highest one to date so far has been about 150 ppms of naphthalene. That has been our highest VOC that we have found. The one problem, though, is the VOCs that we are measuring currently could be lost naturally. If you left the biochar piled out in a pile for a couple months, I would expect to see lower volatiles. So all of a sudden, the VOC signatures that are present on biochar become a function of its storage. And not only, if you remember back that slide I showed for the O to C ratios for the stability of biochar, production is very important. Temperatures drive the primary production of all these chars. However, the storage, what happens to it after that production, is also important. So this explains why in the literature we have a whole variety of stability rates, because not only does it depend on what temperature that char gets to, it's what happens to that char after it's produced, how it's cooled, what processes go into the abiotic oxidation. The O to C ratios capture some of that. The little categories that I showed of the different stabilities do a better job than if I would show just production temperatures or types of pyrolysis or even the material, parent material for the pyrolysis. So that's where I feel that O to C ratio is a very good capture of some of these. Then I've also highlighted some that are currently of very big interest in the biochar community. After this 18-minute time period in this GC mass spec scan, we get into the realm of what I like is the semi-volatiles. These are all of the PAH, the polyaromatic hydrocarbon realm, where we have two benzene rings with a bunch of other stuff hanging off of them. This is the realm of a lot of human carcinogens. They are byproducts of any biomass combustion, and they are present on our chars. You can see the amounts here, and this thermal desorption is not the best method to see these. Because they're semi-volatile, just heating the material up to 50 degrees really isn't an analytical quantification of these PAHs. But we have the suggestion that there are some of these present on the chars in varying concentrations, which probably relates to how the material is stored as well as how it is produced. So overall, all of those volatile compounds, and the macadamia nut char, we have identified 195 different compounds, and we're still not done. The libraries that we need for these haven't fully been developed. Because of the oxygen, we end up with a lot of structural isomers. Once you get 3-4 oxygens on a molecule, you can have 14, almost like 2 to the n number of oxygen isomers. So it ends up in a very complex organic chemistry nightmare. The take-home message is these volatile organic compounds can and do interfere with microbial processes. Terpenoids interfere with nitrification in addition to ethylene, in addition to the alphapinene. The fluorols and derivatives, remember that was a compound that was first suggested by Warnock as one of the important compounds that the biochar is supporting those aiding bacteria to produce. All of a sudden, if we have that absorbed to the biochar, it does that action without needing those helper bacteria. And those compounds inhibit not only microbial fermentation, but also nitrification. One of the little side notes is one of the difficulties in cellulosic ethanol production is this furfural, because it actually inhibits the fermentation process. So we also have benzene and esters present. They are also all known microbial inhibitors. And this issue of these volatiles being microbial inhibitors is a newly emerging research area in the plant and soil dynamic literature. You can see that we're slowly building a knowledge that we need to know how things communicate in our ecosystems. It is often asked by a lot of the first and second graders again, well, how does Mr. Microbe A there know that something is happening? and it is these signaling compounds. So all of a sudden, if we're adding biochar to our soil, not only are we adding new signaling compounds, we are also adding a material that can absorb some of the signaling compounds that they're dependent on for their survival. So overall, this isn't the full piece to the puzzle. I would love to say that I have biochar interaction with soil figured out, but I can't. This is only one little aspect, and ethylene has to be added to our whole suite of mechanisms. The sorbed volatiles and all the degradation products from those volatiles should be included in our mechanisms. They are microbial inhibitors, and it could even help to explain some of the plant effects. The reduction in N2O production has been one of the big banner things that biochar has been cited in the literature. However, now there's two studies, mine and the Tim Close from New Zealand, that are starting to say that it's not the biochar, it is actually these chemical inhibitors. And that's supported because if you look at the activated charcoal in my studies as well as others, there's no impact on the nitrous oxide production. So we're starting to see that, yes, this could be a very vital inhibiting mechanism. The one problem is, as you can see down here, the length of this impact is unknown. How long will these inhibitors act? What length of time are we looking at? We still don't know all of that. So the thing I wanted to send you home with is there's absolutely no absolute biochar trends. Biochar is, remember, the process that we're creating black carbon. Black carbon is a continuum of products. We just don't have a single product in there. We have soots, we have charcoals, and we have chars. All of those are intermixed in what we are referring to as biochar. So it is a highly variable solid material, and the responses to biochar are not only a function of the biochar itself, but also what microbial communities are present. So typically, if I had to be put on the spot, so far the scientific data has shown that it typically reduces the CO2 basal rates, provided that you account for the CO2 evolution from the char itself. It has reduced methane oxidation activity. There's reduced nitrous oxide formation, reduced nitrate production, and there's increased ammonia typically. Exceptions always exist, and they exist wonderfully in the biochar area. So like I mentioned before, I don't have a pyrolysis unit, so I am very grateful to all of the companies that have been able to provide me or link me up with sources for biochar, as well as the USDA ARS Pyrolysis and Biochar Initiative, all the tech support that I get from my technician, as well as the students that I've had working in my lab this summer. The thing I wanted to end with is a quote from FDR. It is, "The nation that destroys its soil destroys itself." I am not saying that biochar is a bad thing. I am saying we need to know what it does to our soil before we apply it universally to all our soils. We are getting into the realm where productive farmland is one of our most precious areas. If all of a sudden we apply a biochar that maybe 10 years down the road could have a potential negative, despite maybe the potential 3-4 years positive growth responses, is it worth destroying that soil when we don't know what it's going to do? So it's one of the people that worked for me said, well, why don't you just go out and sieve the soil to get rid of the biochar? We can't. The particle sizes, it's just not that simple to separate it once it's mixed. So thank you very much, and I'll take any questions.
Are there questions for her?
What about the argument that, say, Johannes made the next slide? The Amazonian soils that have been found in Brazil continue to be extremely fertile, and therefore you can extrapolate. So what about that? What would you say?
The question was regarding, well, how do you actually explain then the terra preta analogy that these are very fertile soils, and why could we have any negative impacts from biochar? Changing some of the words and paraphrasing. My response to that is we're dealing with different materials. It goes back to the whole explanation I had of the natural versus synthetic biochars. The terra preta and all the naturally produced charcoals, they're done in aerobic atmosphere typically, where they might be covered with a little hut or a little wood dome to restrict oxygen, but they haven't purged and flushed it to keep it entirely oxygen free. The other side to it is they just didn't burn one material. There's a lot of archaeological evidence showing that there was clay pots, any other waste materials typically mixed in these fertile areas. Remembering back on that slide that I showed where the natural biochars have impurities, those transition metals, the irons, magnesiums, the coppers, cause catalytic reactions. Putting it simply, you get different things out. We might get more aromatic structure. When Katie showed the diagrams of showing what the chemical similarity of a structural unit of the chars were, we might have in the natural chars higher units. We might have sheets that are approaching full aromaticity where we don't have these oxygen defects. The one thing that should be mentioned is in all activation literature for charcoal, they refer to as surface chemistries. The processes of activation normalize all those surface chemistries. That could be happening over the periods of 1,000 years, where we don't know what happened to these soils after they were added to the soil. They might not have been used for farming, and there's more evidence typically indicating that these were waste piles that weren't their productive areas. They're productive now, but they might not have been in the past. There's a lot of anecdotal evidence pointing to this, where in the Midwest we used to burn corn cobs in our field. You talk to some farmers and they say, oh, I'll never do that again, because they could see the suppressed yields for a couple years after they burned the cobs out there. All of a sudden, 5, 10 years after that burning, that area is productive. So we really don't fundamentally understand all the mechanisms involved in this yet. The question was, what type of biochar is best for applications? And I wish that I had an answer. The difficulty with this is soils are a complex system. Biochars, as you've kind of seen with my little rudimentary introduction, are a very complex system. We have complex squared now. We've been practicing soil science for hundreds of years, and we don't have all the answers for all the soil processes. And yet we're trying to extrapolate what's going to happen to those processes, which we don't fully understand why they're happening, to this new material. So the basic conclusion is we don't have an answer.
As a marketing point of view, ours is the best.
I mean, and that's what one of the little side comment was that the marketing side of it can spin a lot of these things. And that is the real heartwarming thing that I have experienced with this biochar community. The entrepreneurs that are in this field are 100%, all of their effort is behind this material. And I appreciate that. It is very difficult when you're a scientist to try to motivate the community and the public over your research. This has been an absolute simple area. Everyone's excited about biochar, and you don't have to plead with them, well, give me material so I can test it. Everyone's been willing to send materials to test it. So it is a very interesting thing, and that is what's driving this, is all of the entrepreneurs out there pushing and promoting biochar.
In your slide with the continuum from soot up through things like that, you then placed it over the top of it the name biochar.
Yes.
I believe a lot of people in the IBI are saying biochar is the char which is destined for the soil. Now, absolutely any of those things, somebody could throw it on the soil, but let's hope they're not taking some basically really charcoal, tarry filled or something like that and throwing it on. So I find that the expression to be a little bit contradictory to the people trying to define biochar as biochar destined for the soil, which is probably up in the category of more where you have the single word char.
So the question was getting to the whole classification of where biochar is in this black carbon continuum, and about the IBI's efforts of coming up with the standard definition of biochar. This has been a very frustrating issue. I mean, when I first started in all this was back when they started looking at how do we define biochar. Unfortunately, as I've been stressing in my talk here, biochar is not new. We have been creating biochar for 1,500 years at least under industrial processes. The whole thing that's unique with biochar is the fact that we're utilizing biochar as a means or a vehicle for carbon sequestration. That should be the definition of biochar. It is the use of a carbonized biomass for carbon sequestration, period.
In that regard, there are at least several communities dealing with chars and things, all of which comes from the biological material which has been charred. The people are using it for fuel. The people are using it for sequestration. The people who say, I'm all for it for soils. I don't even believe in this global warming stuff, but for soils, that's good. And then there's the people for industrial purposes, which is activated, etc. I understand your position, and I think that this is something that they're going to have to work out a little bit more on the terminology as we go along.
Oh, and I mean, it was another further comment on the overall terminology. And this has been a problem in the coal literature. You look through the coal literature dating back to the 1800s, there's about 24 different name changes for char and charcoal. Science, we're wonderful at classifying things. And it's just been this continuum idea is very difficult for scientists because it is not, you have 0.35, oh, it's this category here. They're quasi divisions. There are so many factors that go into this, and it is a continuum. We don't have breakpoints. We don't have transitions from liquid to solid. We have this entire continuum of one solid material, but it's an entirely different chemical composition. If you look at a chunk of char, if you would do the NMR detailed analysis, all of a sudden you would see variability within a chunk of char. And how do we go about defining a material when the definitions already exist for our solid residual? There's no reason to argue or fight about what the solid residual is. Luckily, the coal people have done that already. What we are promoting as biochar is the sequestration angle, the purpose of why we're creating it. That is our definition, in my view. And I know that's not shared. I'm on all the IBI committees, and you get 14 scientists in the room on this, and you add the industry and all the advocates, you have all different views. And what I am trying to say is everyone should take a step back, look at all the literature that's out there, and realize that we fit into all this. We are not a new material. That was all.
There's some suggestion that biochar will reduce or help with soil erosion. Have you seen that in the literature?
Yeah, I mean, yes, there are. The question was if biochar can aid or improve soil erosion issues. And I always like referring to biochar being an entire panacea. If your soil has an issue, we can come up with a formulation of the biochar or black carbon to improve that issue. There's another scientist in ARS, Jeff Novak, that likes coining the term designer biochars. And I really support that because we're no longer going to be able to go to Home Depot and buy a 5-pound bag of biochar and solve all of the nation's soil problems. It's going to have to be tailored. The difficulty is that tailoring or customization is not fully known yet. I can't give you a recipe for what you're going to need for soil erosion. It has the potential to aid soil erosion as well.
I know you and I have talked about this a lot, but can you comment on kind of the combination of temperature, duration of heating, and kind of particle size as what makes maybe some of the better chars in that temperature, duration, particle size realm?
The question was for me to comment a little bit on the range of temperature, particle size, the processing that actually would go into making a good biochar. My answer to this changes by the week. Right here is my current answer. If you analyze it for an O to C ratio and it's less than 0.2, you have a stable material for carbon sequestration. Now, that doesn't necessarily mean that you're going to have a lot of soil fertility benefit. This is for the carbon sequestration side. The fertility realm is even more complicated, and this isn't even a straightforward answer. So the production is a very vital aspect, but I'm beginning to really think that what happens to it after production could even be equally or more important than the production parameters. And that can be easily shown in all the activated carbon literature, where you'll see, even we've been told today, that the activated carbons are hundreds to thousands of meters squared per gram. The biochars are in the 10s to 15s. There's a lot of conditioning. If you just heat them up to 150 degrees C, you can double that area. If you go a little higher, you might triple the area. And there's been some studies that have shown they can get up to hundreds of meters squared per gram just by conditioning that biochar. So it still needs to be fully worked out, all of these convoluted angles. And I think Kwasi's going to talk a little bit more on the activation and that whole side of it.
One more question.
Okay.
It's a general question. I mean, you told that if we add a lot of biochar in the soils, if you want vegetation and put it in the soil, so after like some period of time, we don't know about it, we have 10 years of pruning. But if that happens, what we have to do the soil to get back the nutrients in the soil.
Okay. So the question was, despite the fact that I view these very short-term impacts of being a negative to all the soil microbial processes, what would happen if 10, 15, 20 years down the road that we actually see overall benefits to the overall soil microbial populations? And yes, that is a very good question. The complication with that is all of our lab studies are an abstraction from reality. When we go out to the field, we take a soil sample, we bring it back, we analyze it. We have already impacted the soil microbial communities. It's been shown that the fungal community, we almost decimate. We don't actually get a good representation of its activity in our lab incubations. So yes, that is a very good point, that we don't fully know what all of the impacts are going to be. That's one of the reasons why within ARS we have this multi-location study looking at these effects at the field plot level. We are currently in the second year of that, and we're slowly getting some results out. But again, we're going to really be waiting for the 5-year, 10-year type time periods to see if we have an actual beneficial impact. So.
Okay, thank you very much.
Thank you.